Namaskar friends. Now let us see that healthcare biotechnology. So what is the use of that biotechnology in the healthcare? The healthcare biotechnology, it refers to the medicinal diagnostic products and the vaccines. Medicinal diagnostic products, that is to diagnose the diseases. The diagnosis is very important in case of that diseases. And the vaccines are produced. The vaccines, we will see what is the meaning of that vaccine. So the healthcare, it refers to the medicinal diagnostic products and the vaccines. And such a field, in that field, here the patient is treated for the organ transplant. Organ is transplanted. Whenever there is a damage of that organ, the organ is transplanted in these cases. The stem cell technology, the stem cell therapy, you know that the stem cell in bone marrow, the stem cells are present, and from that stem cell, the lymphocytes and the immune system, it is related to that stem cell technology. In forensic medicines and the gene probes, you know the probes means the radioactivated single-stranded DNA, which is necessary in case of for the screening of gene library. We have seen that, as well as in case of DNA fingerprinting, here the probes are necessary, so we can produce here the gene probes and the genetic fingerprinting and the karyotyping. So in healthcare, we will see such points. First of all, the human insulin. You know the insulin means. The insulin, it is produced in case of isolate of Langerhans in a pancreas. You know the digestive system of the human being. In that digestive system, the pancreas are present, and the pancreas having a isolate of Langerhans cells. The isolate of Langerhans cells contain the beta cells, delta cells and the alpha cells. Especially the beta cell, it secretes the insulin, and the insulin, it maintains the sugar level in the blood of the human being. And whenever there is a less amount of the insulin, then the patient is suffering by the diabetic mellitus. Patient is suffering by the diabetic mellitus. So diabetic mellitus, it is related to the beta cell of isolate of Langerhans in the digestive system. That digestive system, the isolate of Langerhans cells are present, and when it secretes the insulin, when the insulin level is decreases, the sugar level is not maintained in case of that human being. So such gene, it is isolated. There are the two chains of the A and the B chains. It is isolated from that isolate of Langerhans cells, especially from that B cells, and it is introduced into the plasmid. The plasmid is the vector. You know the plasmid is the vector. It has ability to carry the foreign gene. So in such a plasmid, the chains, these two chains, they are inserted, and in especially the PBR3T2. P means the plasmid, BR, Bolivar and the Rodriguez, 322 are the numericals. In such PBR322, the such insulin gene or the two chains, they are separately inserted in two plasmids. Then these two recombinant DNA, they are inserted or they are cultured, or this recombinant DNA, they are transferred into that E. coli. They are transferred into the E. coli, or they are introduced into that E. coli. And then E. coli, it cultured in a culture medium. This E. coli, now it is able to produce the penicillase and the pre-proinsulin. The pre-proinsulin, from that pre-proinsulin, by the treatment of the trypsin, the insulin is separated. As like this, here the human insulin is produced. Now, the vaccine production. You know the vaccine. Vaccine means it is a heat-killed microorganism or the heat-killed pathogen. The toxins which are produced by the pathogen, they are heat-killed. They are killed, or inactivated toxins, they are introduced into the animal, and it is termed as the vaccines. So the pathogens are here the protein antigen, or the protein antigen. It is inactivated protein antigen which are present on that pathogen. You know the pathogen is here, and on that pathogen, yeah, the antigens are present on the surface of that pathogen. The antigens are present, and it is responsible. It stimulates the immune system. So such inactivated antigen or inactivated toxin material or the heat-killed pathogen, the vaccines are produced. And we have seen in health and diseases, there are the two types of the immunities. Inborn immunity and the acquired immunity. In acquired immunity, the active acquired immunity. In case of active acquired immunity means after the birth, the immunity, in that immunity, after the birth, it is adapted. The person adapts such type of the immunity. Why it is called the active? Because here it can produce the antibody against that pathogen. That's why it is called the active acquired immunity. After the birth, the antibodies are produced. That's why it is called the acquired. But in its own body, the antibodies are produced. That's why it is called the active acquired immunity. In active acquired immunity, such vaccines are produced. So we cannot wait for the natural infection. And for that purpose, here the vaccines are used to stimulate its immunity or the immune system. So such inactivated antigens or such inactivated toxins or the heat-killed pathogens, they are introduced into the animal. For example, the smallpox or the polio. In smallpox or the polio, the vaccines, the heat-killed pathogens, they are given to that patient or given to that organisms to acquire the immunity, to adapt that immunity, to fight against that organism, to fight against that pathogen. In case of that smallpox and the polio, such vaccines are used or such vaccines are produced against that smallpox and the polio. The vaccines are used to eradicate the non-communicable diseases. The communicable diseases, to eradicate such diseases. There are the two types of the vaccines, edible vaccines as well as the melt in the mouth. In case of vaccines, there are the two types, oral vaccines as well as the melt in the mouth vaccines. So these two are the types of the vaccines. The oral vaccines, it is now it is a novel approach, and now it is becoming a hot spot in the case of the vaccination, in case of the vaccine field. The oral vaccine means it is administered orally. So the vaccines are given orally, and that's why it is called the oral vaccine, and it is administered orally. You know that the pathogen, on the surface of pathogen, the immunogen protein is present. Now that is, it is called the or against that here the gene which is present, and it produces the immunogen protein. And whenever it is activated, when we administered orally, such gene or when such pathogen it is administered orally, it becomes activated and it produces the diseases. So such gene which is responsible for that disease, and whenever it is administered orally, then such vaccine is called the oral vaccine. Now in case of that, here the such gene or which produces the diseases, such a gene, it is isolated, and this isolated gene, it is introduced into the plasmid. When such gene or any gene, foreign gene introduced into the plasmid, then it is produced the recombinant DNA. We know that the recombinant DNA, it is a manipulation of the gene from that pathogen and the plasmid from that bacteria. When it get recombined, the recombinant DNA is produced, and such a recombinant DNA, it is introduced into the plant, a part of plant. In case of stem, in case of leaf, or in case of fruit, here it is introduced. And when such a plant or the part of the plant, when fed to the animal, especially the human being, the patient is termed as the vaccinated patient. And such type of the vaccine is called the edible vaccine. Such type of vaccine is called the edible vaccine. That means the gene which is responsible for that diseases, or the antigen which is responsible for that diseases, or such is, it is called the immune protein which is present on that pathogen. When it administered orally, it get activated. So such type of gene, when we isolate from that pathogen and then it introduced into the recombinant DNA or introduced into the plasmid, then it produces the recombinant DNA with the help of the enzyme, endonuclease enzyme and the ligase enzyme. The recombinant DNA is produced, and such a recombinant DNA, when it introduced into any bacteria, and such bacteria when we introduce or when we insert in a plant part or in a plant, whole plant, and such plant when fed to that animal, the such animal or that human being is called the vaccinated. The person is vaccinated. That is, the vaccine is given to that person, and then their immune system is stimulated to that person, and the antibodies, now the person's immunity is able to produce the antibodies. Okay, so this is called as the edible vaccine. Nowadays, the edible vaccine can be produced with the help of that biotechnology. That is, the plant part which is edible, or the plant which one is the edible, in that plant the gene is introduced. The gene which stimulates the organism to cause that disease. When such gene is introduced in the plant with the help of that recombinant DNA technology or with the help of that biotechnology, then it is called the edible vaccines. Suppose the banana which having some vaccines, the gene edible or the pathogenic gene which is termed as a vaccinated or the edible vaccine. In case of apple, there may be the possibility to produce the edible vaccines in case of apple, in case of banana, in case of custard apple, whatever it may be. Okay, then now here the melt in the mouth vaccines. That is, when the vaccine or this gene, it is administered by placing under your tongue, then it gets activated and it enters, it is delivered into the bloodstream, and the patient produces the antibodies against that pathogen. So for example, the vaccines against that flu, or the flu vaccines, and it is produced by the bacillus bacteria. Such bacteria, whenever we can place under your tongue, then it acts as a vaccination. And what is the benefit for that melt in the mouth? The benefit is it is a very comfort for the administration. It having a low cost, and it is easy for the storage. So these are the two types of the vaccine. Nowadays it is a very important in case of that vaccination field. It is becoming a hotspot as well, in case of the vaccination, and it is having a novel approach in case of that vaccination field. So there are the two types. Which are these two types? That is the edible vaccines and the melt in the mouth. Edible vaccine, that is the plant part. Which one is the edible? In that plant part, the gene responsible for that disease, it is introduced or it can be inserted, and we can eat such a part which having the recombinant DNA, and it act as a vaccines. So we can produce the antibodies against that pathogens. And melt in the mouth, that is the bacteria or the vaccines, when such antigen or that such pathogen, when we put or the place under the tongue, it can be melt. That's why it is called the melt in the mouth. It goes or it deliver into the blood. It deliver into the bloodstream, and it produces the antibodies. Nowadays, here the few vaccines can be produced, or the few vaccines can be produced, and with the help of that bacillus bacteria, and we can produce the antibodies against that flu. And what is the benefit of that melt in the mouth? It having a low cost, as well as the it is easy to store, and it is comfort for the delivering. So these are the different points we have seen in the healthcare, as well as the genetically modified plants we can produce with the help of that biotechnology.